Hey everybody, and welcome to Lab 4 of the Neuroanatomy Lab series. Today we're going to be talking about the thalamus and the basal ganglia mostly, and we will be looking at those structures on both a human brain model and a thalamus model, but as well as doing an actual dissection with the sheep brain. So it'll be a little bit similar to the first lab you went through where we did a sheep brain dissection, but instead of making a mid-sagittal cut to start and coronal cuts, we're going to make axial cuts and kind of get to these deep brain structures. And I'll kind of show you what type of cuts we're going to make as we go through it with the human models, and then we'll go ahead and jump into the dissection. So in your lab manual, the first couple of slides are just kind of like the organization of the lab as it normally is. But really what I want to talk about first are the basal ganglia and thalamus structures. So I'm going to focus in on this human brain model here. And if we were to remove parts of the cerebrum, you can see some of these structures a little bit underneath. So we're going to kind of hone into these guys here. So I'm going to pull them out so you can see them a little bit better. So here you can see the lateral ventricles. We're really going to keep making a bunch of cuts on the actual sheep brain until we can start to see this opening. Right here they have it as like an actual structure, but they're essentially holes in the brain where you have cerebrospinal fluid. So we're going to cut down until we get to that, and we'll start to see the hippocampus kind of peek through there, and that's essentially what we're going to dissect out. So I'm going to go ahead and kind of pull this out here a little bit more so we can talk about it. So I'm going to kind of remove cerebellum and more of these structures. All right. So more or less, these are kind of the structures we're going to get to, and I'll remove the insular cortex here so you can kind of see some of these structures more specifically. So the first structure that we kind of are going to show in the actual sheep brain today is going to be the basal ganglia. So you can see some of the structures of the basal ganglia here. So here is the caudate, and right here is the putamen. So the basal ganglia is essentially involved in movement disorders and permitting movement and inhibiting movement and so forth. And you probably hear it when you talk about things like Parkinson's disease and Huntington's disease in the 101 series. If you've taken 101A yet, you'll talk about it more extensively there and a little bit maybe in lecture. But when they refer to the striatum in those disorders, they're really referring to the caudate and the putamen. And I'll kind of go through why they get that name in a little bit. But something to keep in mind as you go through the lab and also the lecture is that even though the basal ganglia is mostly talked about in terms of movement, they're also implicated in other stuff as well, such as reward circuitry, motivation, as well as emotions. And they have a lot of different cortical basal ganglia loops between the cortex and the basal ganglia that are important for a lot of those functions. And a lot of that research is still ongoing, so something to keep in mind. The thalamus is actually located a little bit medial and caudal to the basal ganglia. So here are those basal ganglia structures, and then in here is actually the thalamus. And you'll kind of see that a little bit more on the sheep brain and when we kind of take some of this stuff apart, and you'll also see it on the human brain models next week. The thalamus is essentially involved as a sensory relay, which you probably know from your undergrad bio classes, where it's essentially relaying sensory information to the cortex. Well, that's kind of its broad function. There's other functions as well. And the thalamus has been implicated in consciousness and arousal. So lesions to the thalamus can cause deficits essentially in consciousness, something to just kind of keep in mind going forward. And the hippocampus is actually on this little piece that we removed. So this kind of sits in here, right? This structure here is the hippocampus. I have a pencil here that would probably be better than my finger. But this structure here is the hippocampus, and you can kind of see that when we cut through essentially the ventricles, you'll start to see the hippocampus in the actual sheep brain. The hippocampus is involved in spatial navigation. It's also involved in learning and memory, which is most notable for. But there's actually differences between the dorsal hippocampus and the ventral hippocampus. The ventral hippocampus has been implicated in things like anxiety and feeding behaviors. That's something you'll learn about a little bit more in 101C, or have learned if you've already taken it. So a lot of these basal ganglia structures actually have this characteristic C shape. That's due to the telencephalon actually folding over the diencephalon, which is essentially the top of the brainstem here, including things like the thalamus and the hypothalamus. And so a lot of the basal ganglia structures, as well as the ventricles and the hippocampus, have that kind of C shape. So just keep in mind that the reason it's like that is because of the telencephalon folding over. Something that you should keep in mind with the basal ganglia itself, it usually gets referred to by a lot of different names, one of which is the striatum. The reason it has that name is because if you look at the caudate, which is this top part that wraps around, and the putamen, which is this middle part here, there's these little striations that connect the two. And these white matter fibers that you can see kind of peeking out that are coming through, that's the internal capsule. So if you were to look at this in a section, like a coronal section, like we saw during week 1, during the sheep brain dissection, you would actually see some of these striations, particularly if you had a stained specimen. So whenever you hear striatum, they're just referring to collectively the caudate and the putamen together. So if I pull this side off, they're actually showing the full internal capsule coming through here. So this is the putamen, and now you can't see the caudate because of the fibers that they're showing kind of coming through. On this side, they're just showing them kind of cut away. Also, there's another name that you should be familiar with, the nucleus accumbens, which is essentially where the caudate and the putamen meet. The nucleus accumbens is really important for reward circuitry, and you'll hear a lot about it with regards to addiction in 101C. The globus pallidus, which is just medial to the putamen, it's kind of just on the inside, that has a lateral and a medial segment. Sometimes it's referred to as the globus pallidus externa, or external segment, and the globus pallidus interna, or internal segment. Collectively, some papers refer to the putamen and the globus pallidus together because they have a similar shape, and when they do that, they just refer to it as the lentiform nucleus. So again, in your lab slides, there's a bunch of different cross-sections of brains where a lot of these different structures are kind of labeled. So you can see how it would look in different vantage points in fMRI or MRI or actual brain cuts like axial cuts or coronal cuts. So just be aware of where these structures are in those different cuts as in the lab manual and be able to identify them. If you were given it on the exam, like on the practicum or on a quiz, we would give you the exact images that you see in the lab manual. Another thing to kind of keep in mind about the internal capsule, I kind of mentioned it a little bit earlier. These are the fibers that are essentially going to and from the cortex. So this would come out into the corona radiata and connect with the cerebral cortex itself. But essentially, it's just containing the ascending and descending fibers that need to communicate with it. There are stops with the actual thalamus in the internal capsule. So some of these fibers come up, synapse in one of the nuclei of the thalamus, and then it gets relayed from the thalamus out through the internal capsule to the cortex. There's a nice video on your lecture slides. There's a YouTube link, and that actually shows you a 3D computer model that shows the internal capsule kind of coming between these structures really nicely, which I highly recommend essentially checking out. Okay, so there's a couple of slides that essentially show you more or less what I talked about. I'm actually going to put this back together briefly. I'll do that later. Just going to put this off to the side for a second. And now we are going to focus on the thalamus. So this model over here is essentially the basal ganglia along with the thalamus and the hypothalamus. We're going to just focus on the thalamus, so I'm going to remove it here. So this structure essentially is also shown in your lab manual. And there's a bunch of different nuclei of the thalamus that you're going to talk about in lecture. And in lecture, you need to know the functions. For the lab purposes, you just need to be able to identify them on this model. So there's a couple of different nuclei that you should be able to identify. The first is the anterior nucleus. This is this guy right in the front. So anterior, right? So this would be essentially the nose. And this is the anterior-most portion of the thalamus. So it's called the anterior nucleus. Its function, in case you were interested, is actually one of the relay stations for memory. You actually have tracks that are coming from the mammillary bodies, one of the nuclei of the hypothalamus that come and synapse in the anterior nucleus through the mammillothalamic tract, something that we'll talk about a little bit later. Then there's the ventral anterior nucleus, which is this guy right here. The ventral lateral nucleus, both of which are essentially involved in motor behavior. So there's synapses here that are involved in a lot of the different motor areas of the brain. Then there's the ventroposterolateral nucleus, which is a sensory relay station. And there's also another nucleus called the ventroposteromedial nucleus, the other half of it, which you can actually just see on the inside here once we take it apart. Then this guy back here is the pulvinar, and then up here we have the lateral dorsal nucleus, and then here we have the medial nucleus. So these are all the ones that you need to identify on the lab practicum or the lab quiz, and you're straight up going to get just the image that's in your lab manual. So just be able to identify them there, and you should be solid. Note, for your own purposes, there are 2 nuclei of the thalamus that are not represented on this model. Do you know which ones they are? Well, they're the medial geniculate nucleus and the lateral geniculate nucleus. Those are relay stations in vision and auditory information. And you actually can't see them on this model. I think they're actually on this one back here. But keep in mind that those ones are not on here. So if you blank on the quiz or the practicum, just remember that those 2 are not present. And with that, we are going to go ahead and go over to the actual dissection with a sheep brain. Okay, so now we have our sheep brain, and we're going to go ahead and start doing the thalamus and basal ganglia dissection. If you're following along at home and you have your own sheep brain, you're essentially going to want to wear proper PPE. So notice that I've got gloves. I cleared the table of any models that I do not want anywhere near something like a specimen. I got goggles, my lab coat, and closed-toed shoes, long pants, that sort of thing. I do have this laptop here. But it's an old laptop. I really don't care what happens to it. Throughout this dissection, I'm also going to invite a cameraman who's also in the vicinity to get really close, so hopefully you can see very fine details of the structures that we want to actually see. So the first thing you're going to do, you can follow along in your lab manual, is we're going to essentially make cuts from the dorsal surface, and we're going to make axial cuts. So we're going to essentially cut like this and keep cutting down until we start to get to where the white matter, again, those axons kind of start to fan out into a sheet. That means we're getting really close to the lateral ventricles. So I'm going to go ahead and start shaving off parts of the cortex. So I usually don't see it until I start to get to where the cerebellum is. So for the first couple of cuts, you can be pretty liberal. So I'm going to go ahead and shave some of these. So you can see here that we didn't quite reach it yet. So the white matter is again in here, and you can't really see any kind of fanning out into a sheet, so we need to keep cutting. You want to try and be as consistent on both sides as possible because it's really easy to trim one of the hippocampi. So you following me, camera guy? Because this is about to get furious. So we're going to keep shaving. Hopefully if you're at home, you have a sharper knife than the one I have here. So I am just starting to potentially see it, so I want to be kind of careful here. So I can kind of see it on this side, right? This little cavity that starts to form here, that is the lateral ventricle. I want to get till I see it on the other side too. Doesn't matter if you kind of mess up the cortex, it's not important at this point. For this dissection anyways. Okay, so we should start to see it on this side too. Let me see if I can dig in here a little bit and see if I can see it. Not quite yet. So now you can kind of see it on both sides, right? So right here you can start to see the hippocampus just peeking out. So we've cut right through the lateral ventricle here, so you can kind of start to see it. So what I'm going to do is now I'm going to kind of wedge in here so I can shave off cortex. So what I'm going to do, so the hippocampus is in here and it kind of wraps around like this. So we're going to try and cut this way so we can remove this cortex on the sides and kind of like dig out the hippocampus. So I'm going to cut this way a little bit. So I'm going to do the same on here on this side. So this stuff here looks like choroid plexus to me. Those filtering blood to make cerebrospinal fluid. So let me sorry, Jeremy, it might be hard to get some of this stuff because I'm trying not to botch it.
Yeah, it's all good.
So you can start to pull a little bit and cut some of this cortex away. So we can start to dig it out. So I'm going to try and gently cut through here. If you're using a knife at home, be very careful depending on how sharp it is. This is pretty dull, so there's not really a risk of me injuring myself. Nope. I also need to dig it out on this side. Here, this might also help to cut up. So here you can kind of see the hippocampus right here. So this is your hippocampus kind of wrapping around, and this is where the fibers come together to eventually form the fornix, and they kind of go down in here. If you peek into this cavity, it's the lateral ventricle, you can see the caudate head, these bumps on either side. Now I've cut into the caudate here, but you can see where they are if you peek down. Try and remove some of this so you can kind of see. And then again, here you can see the corpus callosum those white matter fibers going across to either side. So I'm going to try and some of this stuff. I want to see that pineal gland. So in your lab manual, you'll kind of get to something like this. So sometimes you'll see the name cornu-aminis, which is, or CA. Sometimes you'll see the different hippocampus regions that are referred to as, CA1, CA3, and so forth. That essentially comes from the old name Horn of Amun, the Egyptian god that had a ram's head. So you can see kind of the ram's horns of what the hippocampus looked like, right? So if you were to look at it, it looks kind of like a ram's horn facing this way. Some other structures from previous labs we can actually see here. So you can see the pineal gland right here, this little bump here. And you can see the tectum, the big two superior colliculi here, and the inferior colliculi just below it. So again, like on the human brain model, we essentially have the caudate, right? And then just underneath the hippocampus is going to be the thalamus. So what I'm going to do now is I'm going to essentially peel the hippocampus this way and show you the thalamus underneath. So in the lab manual, you will essentially see plate 6. That's what I'm going to do now. So I'm going to pull this up. So I'm pulling the hippocampus up. All right. And then these two bumps here are the thalamus. And again, here's the pineal gland and the superior colliculi. So another thing that we may want to do is actually remove the cerebellum so you can see into the fourth ventricle. So I'm going to do that now. So essentially you'll want to very carefully cut away, and I'm going to pull down and pull it like this. So we're pulling the cerebellum off. I'm going to try and clean it up just a little bit. So one thing that we talk about in the lecture, if you've gotten to it by the time you're watching this video, is the cerebellar peduncles. So the cerebellar peduncles are essentially feeding information to and from the cerebellum. So the cerebellum is just sitting like that. And here are the pons. These actually wrap around to form the middle cerebellar peduncle, which feed into the cerebellum itself. And I've essentially cut that across. And then the fourth ventricle, again, is right underneath where the cerebellum was. So this is essentially what you want to get to in your dissection if you do it at home. Okay, so just to recap, we cut down, made axial cuts until we got to the lateral ventricles. Then we peeked in and saw the hippocampus, careful not to cut through the top of the hippocampus there. Then we kind of dug the cerebrum out to kind of get at these, this hippocampus as well as some of these deep brain structures. and now we can see the caudate on both sides. The hippocampus, they come together to eventually, they have these fibers that come out that form the fimbria, and they come together to form the fornix. Again, that's going all the way down, and it'll actually send projections to the mammillary bodies. You can see the pineal gland, the tectum superior and inferior colliculi, which is review, and then we cut the cerebellum so that we could see the cerebellar peduncles. Okay, so that concludes the sheep brain dissection for the thalamus, basal ganglia, and so forth. So review everything that's in your lab manual, go through the structures. We will give you the exact pictures that are in the lab manual, not what was shown here, so that you can study that and essentially get prepared for the lab quiz or the lab practicum. If you have any questions at any point in time, feel free to contact us on the discussion forum or in office hours. So thanks for watching.